Let's begin discussing neurophysiology. We're going to begin by describing the three roles of the nervous system, and we're going to describe it by using three representative neurons that represent each of these different roles. So in light blue, we're drawing a sensory neuron responsible for sensing things from within and outside the body. In green, we draw an interneuron, which is responsible for integrating information and making decisions. And then in red, we're drawing a motor neuron, which is responsible for effecting responses to whatever was sensed in the first place. So an easy way to describe this would be to use a standard reflex response. For example, if a sensory neuron were to sense a flame and the finger got a little too close to that flame, the body would respond by making a decision in the interneuron level to send a command to the motor neuron to pull the finger away. So these stripes that you see there would be the striations of some kind of a skeletal muscle which would be involved in this movement. Now this looks a lot like a homeostatic feedback mechanism, and in fact it reflects exactly what we've studied in the past with homeostatic feedback mechanisms. You have a receptor, an afferent pathway, a control center, an integrator, an efferent pathway, and then we have the effector and then the response. So it's actually reflecting exactly that situation, and I kind of drew it that way on purpose to make sure that you were able to see the similarity there. Now, this dotted line here represents the division between the two major branches of the nervous system. I'm just going to write the letters because we don't have a lot of space. CNS and PNS. CNS meaning central nervous system, which includes the brain and the spinal cord. PNS meaning peripheral nervous system, which means all of the structures outside of the central nervous system. There's the sensory branch, and then there's the motor branch, and then you should know all of the subdivisions below them. For example, the sensory branch has visceral and somatic branches to it, and then the motor division has autonomic and somatic, and the autonomic branch is further divided into parasympathetic and sympathetic branches of the ANS. So we will be, basically the rest of the course, at least this semester, is going to be focusing on all of the details of all these little bits of the nervous system. Now we're going to go ahead and draw something that we've actually drawn in the past. Those little orange structures there represent some cell membrane, some plasma membrane. And you may recall this structure here, which is a receptor. You see the little binding site there? So the receptor is a big portion of what we're going to be discussing today. It's quite important there's a reason why I emphasize this early in the course and continue to discuss receptors. They are proteins often embedded in membranes that allow us to receive information. So it's quite easy to see how it is that a sensory neuron, of course, would be able to have a receptor. For example, you'd have to be able to understand that you are touching something like heat or a touch sensation or smelling something. I think the idea of receptors is fairly easy in that instance. And by the way, sensory neurons are just amazing and special. We will be talking about them down the line. But what might be a little harder to understand is that there are going to be receptors at various other levels of this diagram. So for example, the interneuron in the central nervous system needs to be able to hear what the sensory neuron is telling it, so it needs to be able to receive that information. The motor neuron needs to be able to receive information from the interneuron, and there have to be receptors additionally at the level of the muscle itself in order to receive the information from the motor neuron. So I've found that this is the most challenging concept for students to get across, is that connection between one neuron to the next to the next. There's got to be a receptor there. We're going to be talking quite a lot about them. So now what we're going to do is we're going to go ahead and name the different parts of a kind of a standard generalized neuron, a multipolar neuron, and we're going to draw two of them because one neuron by itself doesn't mean much. Dendrites, cell body, axon, axon terminals. Let's go ahead and draw our second guy here. Dendrites, cell body. Sometimes it's helpful just to go ahead and draw those. The axon, and right away draw some myelin sheaths. because almost every neuron has some level of myelin along its axon and then the axon terminals there. So let's go ahead and label those. First of all, we'll draw our myelin sheaths, label that. There's two different ways that myelin exists. The myelin sheaths could be produced by Schwann cells in the peripheral nervous system, or they could be produced by oligodendrocytes in the central nervous system. But in either case, they basically work the same way to insulate a portion of the axon with gaps between for neural transmission to occur, and it makes neural transmission faster. So we labeled our axon terminal, which does have, our axon terminals do have other alternate names, but I just use axon terminal. It makes the most sense. The very first part of the axon is the axon hillock. Mind the spelling. The axon hillock is perhaps the most important part of the neuron. We'll talk about why. Then we have the cell body, which is also called the soma, S-O-M-A, and then the dendrites, which are the receivers of information. We're typically thought of as the receivers of information. Now, information, the neural transmission you probably know about is called an action potential, and an action potential makes its way only one direction from cell body to axon terminal down the length of the axon. But there's lots of other types of neural transmission that are also important, such as the reception of information, which we will be talking about more today. Lastly, the last structure we'd like to pay attention to is called the synapse, which is a small space between the cells, which allows for information to be passed from one cell to the next. So the cell that is cell number one, the one that is talking, is called the presynaptic neuron because it comes before the synapse. And then we have the postsynaptic neuron, which receives the information at the level of the synapse. I'm going to put an orange box around some structures here because I want to emphasize the portion of the neuron which is receptive to signal. In other words, which portion of the neuron actually has these proteins called receptors. And so we find receptors, of course, in the dendrites, which is typically what we think of as receiving information. And then we also have receptors on the soma, the cell body. And we have receptors, it turns out, on the axon hillock. That's actually a really important role as well. This is one of the reasons why the axon hillock is so special. That very first part of the axon is quite special and actually has receptors for information and can hear stuff from other neurons. And of course, another box goes to the postsynaptic neuron because exactly the same things apply, no matter if it is a neuron that is sensitive to something happening outside of the body, such as a sensory neuron, or if it's an integrator, which is listening to the sensory neuron, or if it is a motor neuron, which is listening to the integrator. You have to understand that neurons are always set up like this. You have a receptive area, and what makes it a receptive area is that it has receptors. Right, so now we're going to start to draw a cell membrane. It's been a little while since we've drawn one of these, so let's go ahead and draw our phosphate heads in blue and our fatty acid tails in orange. Recall that the outside of the cell is portrayed up above, and we're going to label this IF for interstitial fluid. And the inside of the cell is portrayed below. We will label this as ICF for intracellular fluid. The first thing I'm going to draw here is on the inside of the cell, we're going to draw a number of different negatively charged entities. Mostly this would be proteins and phosphates, negatively charged. And what happens is, and this is important for later, some of these negatively charged structures are going to line up on the inside portion of the cell membrane. The next thing we're going to do is draw some sodium ions. You may recall that sodium ions, which are positively charged, importantly positively charged, exist in higher concentration outside of the cell and in lower concentration inside of the cell, which is the purview of the sodium-potassium pump, which enforces this gradient, this difference from inside to out. So a large number of these Na pluses, these little pink dots outside of the cell, and a small number inside of the cell to understand that there are some inside of there. But most importantly, we're going to go ahead and line up some of these positively charged sodium ions directly on the outside of the membrane. The reason they're lined up there is because they are attracted across the membrane to those negatively charged entities inside the membrane. So I'm continuing the story of this cell membrane, and I'm going to draw, first of all, a channel, and then I'm going to continue the membrane. You'll see that there's kind of a curve down of the membrane because we're going to be drawing mechanoreceptors, which are receiving touch. It's the simplest way that we receive information. So this dotted line out here, let us say that this is a touch, something touching the skin, for example, and eventually pressing down in such a way that some kind of a sensitive cell will receive that touch information. The way that it works is, let's picture this as a tactile cell in the stratum basale. You know that these are very close to the surface of the skin, so any kind of touch there would be fairly immediately understood by those tactile cells. So the tactile cells will have these types of receptors, mechanoreceptors, which are going to be dotted along its cell surface and sensitive to touch. So what happens is, on the left-hand side, we have a mechanoreceptor which is closed, and on the right-hand side, we have a mechanoreceptor which has been pushed open, kind of gaping open in this way. Just kind of squashed. It's quite simple. It's very straightforward. It's kind of squat. I don't know if I'm portraying it well, but you get the idea as I describe it, hopefully. So the actual pressure from outside of this cell will push open the channel, you see? So now it's important to understand the ion that's involved in this. The reason we're kind of focusing on sodium ions in the first place is that sodium ions oftentimes are the instigators of most every neural impulse, and this would be one of them. So what we have, just like we portrayed earlier, we have a large number of sodium ions outside of the cell and a small number of them inside of the cell. So there's that potential. The word potential is quite important. There's the potential for sodium ions to enter the cell, and they really want to. First of all, they're attracted to those negative entities inside, and then they also are attracted to an area where they are less concentrated. Simple diffusion, right? Things pass from an area where they're highly concentrated to an area where they are less concentrated. So let's go ahead and portray all those negatively charged entities lined up along the inside of the cell. So just to really push home the idea that yes, we have this negatively charged interior that the sodium ions are quite attracted to, and so it's very easy for them to pass through the channel as soon as they are given the opportunity. First of all, they're basically lined up. If you want to personify this, they're basically chomping at the bit to get into the cell through their diffusion gradient, passing from an area of high concentration to low concentration, and passing into an area where this positively charged ion is attracted to negatively charged structures. This is our first example of the details of how reception actually works, and it pretty much always happens this way with mechanoreceptors. It's generally what we call the influx of sodium ions. When sodium ions influx or enter the cell, they raise the membrane potential and cause excitation. Now that center part of the statement you might not quite understand yet. We haven't quite discussed membrane potentials, but the idea of being an exciting presence, basically a positively charged structure flooding into this cell, is something that I think would be relatively easy to grasp. So a sodium ion positively charged, as it floods into a cell, it's going to cause more positivity, more excitation. Right, let's go ahead and draw our potassium story, so Ca2 Plus. We will draw lots of these, and I'm going to redraw these with a darker green in a moment because it's just not really showing up. So potassium Ca2 Plus is in higher concentration inside of the cell and in lower concentration outside of the cell. Again, this is a gradient which is ensured by the presence of the sodium-potassium pump. We'll also go ahead and draw chloride ions, which you'll notice this is our first ion that we're describing which has a negative charge. This is going to be important in a moment. We did have proteins and and phosphates inside of the cell, of course, that are negatively charged. But this is the ion that we discussed that is negatively charged. So chloride ions are in a low proportion inside of the cell and a relatively higher proportion inside of the cell, outside of the cell. So when given the opportunity, chloride ions will influx. And I did forget to say, when given the opportunity, potassium ions will efflux. And this just has to do with their concentration gradients, their diffusion gradients. Where a structure is more highly concentrated, it will tend to flow to an area where it is less concentrated when given the chance to do so. Just filling in those greens just a little bit darker to a little more visibility. Okay, the last ion that we're going to be discussing is calcium, Ca2 Plus. It turns out that calcium is a special ion that does not exist inside of the cell pretty much at all. So calcium exists in about 1,000-fold more concentration outside of the cell. This is our highest gradient. This is our greatest difference from outside to in. It's not possible for calcium to enter the cell, except of course when a channel opens up for it to do so. And when that happens, it's extremely powerful. The calcium absolutely rushes in, and then calcium makes things happen. The reason why calcium does not exist inside of cells, outside of like membranous vesicles where it is protected inside of there, the reason it doesn't exist is that calcium is special. Keep that in mind. We'll be discussing it more down the line, plenty. So we discussed the mechanoreceptor, which is a particular way to receive information about touch. Just a simple influx of sodium ions excites the inside of a neuron. But I would say the receptor that is most well known is probably the ligand-gated channel, also called the chemically gated channel. I'm going to say this right off. When we talk about a ligand or a chemical, what we're actually discussing are neurotransmitters. So when a neurotransmitter binds to one of these binding sites, causes the channel to pop open and ions to flux one way or the other, either an efflux or an influx, depending, of course, on where the concentration gradient is higher or lower. It's important to note at the start that the ligand, the chemical, the neurotransmitter, whatever you want to call it, will be binding to the outside of the cell. It cannot flow in. The ion that is flowing cannot bind to the binding site. These are two separate roles. Oftentimes they sort of get suffused together in people's minds and becomes kind of a misunderstanding. So the neurotransmitter binds to the binding site, and as a result, the channel opens up and ions flux through. I'm going to draw two different sets of these. We're just taking a small piece of the plasma membrane and chunking it out, and we're going to give two of the most straightforward examples of how this can work. So first of all, we have one particular chemically gated channel, ligand-gated channel, which is permeable pretty much only to sodium ions. And so we're only going to draw sodium ions, of course, greater concentration outside of the cell. I'm not even going to draw the ones inside of the cell. We're going to go ahead and draw them lined up as sodium ions are wont to do, although we're not drawing the negatively. We've done this enough times, you understand there's negative charges on the inside. Kind of ran out of room, so I had to put my key way up here. So the first type of neurotransmitter we're going to draw is acetylcholine, which is abbreviated big A, big C, small h. Acetylcholine in this context is considered a neurotransmitter that has a really simple role. It binds to this particular ligand here. Look at that. It's binding to the binding site there. And as a result, as you see, the channel has opened. And in this case, this is a channel that is permeable to sodium ions. So these positively charged sodium ions rush into the cell in the direction of its diffusion gradient and its electrical gradient, where it's negatively charged structure inside. And therefore, because sodium ions, these positively charged ions, are rushing into the cell as a result of this acetylcholine binding, acetylcholine is considered to be an excitatory neurotransmitter. Acetylcholine actually has other roles, which we'll be discussing down the line, but in this context, it's super straightforward. Okay, our next neurotransmitter we're going to be considering is Gaba, G-A-B-A, which stands for gamma-aminobutyric acid. You never have to know that. It is considered an inhibitory neurotransmitter, and why? So here, for this membrane, we're going to draw our GABA floating around outside of the cell, binding to the binding site, and in this case, we are going to take chloride ions. Remember, they're negatively charged, and they're in slightly higher concentration outside of the cell than inside of the cell. So let's go ahead and draw that. Chloride ions have a diffusion gradient, which means that when given the opportunity, they will influx. And there we go. Gaba, gamma-aminobutyric acid, is a neurotransmitter that binds to chloride ion channels and therefore allows for the influx of chloride ions and therefore brings a negatively charged structure into the cell, therefore depressing the cell. It really is as simple as that. Positively charged things will excite the cell, like sodium ions. Negatively charged structures, such as chloride ions, will depress the cell, will inhibit the activity of the cell, and therefore this opposing role of acetylcholine and Gaba is very useful. It turns out these are the primary neurotransmitters that oftentimes are responsible for either excitation or inhibition of neurons.